Hello students, in this video we will continue with osmotic experiment. We will finish osmotic experiment. It is we have seen, consider this is as semi-permeable membrane. Solution A, solution B. We have these are solvent of solution B. This is solute molecule. We have too much solute molecule in solution B. Osmotic process is nothing but solute molecule from solution A will move towards the solution B because solution B has high concentration of solute molecules. So the solvent molecules from solution A will move towards solution B because the solute molecules in solution B is greater than the solute molecule in solution A. The pressure that resists the flow of this solvent molecule from one medium to another medium during the osmotic process. This pressure is exerted in this medium. This pressure is known as osmotic pressure. Osmotic pressure. What is osmotic pressure? Pressure that resists the movement of solvent molecule from one environment to another environment during the process of osmosis is known as osmotic pressure. Okay. Is defined as pressure required. Pressure that is required to stop the entry of water. Pressure that is required to stop the entry of water through the semi-permeable membrane. Now we will see about tonicity. What is tonicity? Tonicity is nothing but measure of osmotic pressure gradient. We have seen what is osmotic pressure. Measure of osmotic pressure gradient. That is, if you keep a cell inside a solute, how the cell responds to the solute, how the solvent responds to the cell, what kind of pressure is being experienced by this cell, what the cell will undergo if the cell is placed in a hypertonic solution, in hypotonic solution, in isotonic solution. What are the meaning of all these three? We will see now. First we have seen what are the three types of tonicity. That is hypertonic, hypotonic and isotonic. What is hypertonic? We will see what is hypertonic. Hypertonic is nothing but a solution that has higher solute content than another solution is called as hypertonic solution. Solution that has higher solute content. Now we will see about what is hypotonic solution. Is little opposite, exact opposite. What is hypertonic solution? Hypotonic, a solution that has lower solute content is called as hypotonic solution. Okay. Then what is Isotonic solution. What is isotonic solution? Solution that has same solute content as another. That has same solute content as another. Hyper means high. Hypo means low. Iso means the same. These are the meaning of these three terminologies. Iso means same, hyper means high solute, hypo means low solute content. We will see about various cases. Okay. When a plant cell, assume this as a plant cell. When a plant cell is placed in a hypotonic solution, what happens? When a plant cell is placed in a hypotonic solution, what happens? Let's see. So let's place a plant cell. Assume this as an hypotonic solution. What happens? So hypotonic solution. Water will enter into the cell protoplast and cell become turgid. New terminology, that is, what is turgidity? What is turgid? When a cell is placed in a hypotonic solution. Hypotonic solution is nothing but a solution which has very low solute concentration. When a cell is placed inside it, the solute will enter into the cell. And this content that enters into the cell, that exerts the pressure on the wall of the cell. This wall of a cell will experience some pressure. That is called as turgor pressure, and the cell become bulged. That is called as turgid. The cell become turgid. Okay, this is the first case. When plant cell is placed inside a hypotonic solution, solvent content that in the hypotonic solution will enter into the protoplast of the cell, that is into the protoplast, cell protoplast, and it exerts a pressure. The cell become bulged. This is called as turgid. This pressure experience is called as turgor pressure. It's called as turgor pressure. This is the first case. Okay. Second case. When a plant cell is placed in an isotonic solution, what will happen? What will be the net movement of water? The net movement of water is net water movement is equal to 0. There will be no net movement of water since the solvent is isotonic in nature. Third case, we are going to see about when a plant cell is placed in an hypertonic solution. What is hypertonic solution? The solution that has high solute content. Plant cell is placed in the hypertonic solution. Let's see. Beaker. Let's draw a plant cell. Let's add some hypertonic solution. Consider this blue lines as hypertonic solution. So what will happen if a plant cell is placed in an hypertonic solution? Water will move out of the protoplast because the concentration of solute outside the plant cell is very high when compared to the concentration inside the cell. So water inside the cell will move out of the cell. So the cell shrinkage will occur. Cell will shrink. All the water will move out of the cell. Size of the cell decreases and they shrink when they are placed in the hypertonic solution. Cell will become flaccid. Two terminologies, new terminologies. One is flaccid, other is shrinkage with regard to hypertonic solution. Let's see about few other terminologies. So we have seen what is turgor pressure. Turgor pressure is associated with when a plant cell is placed in an hypotonic solution. Flaccid is associated with hypertonic solution. Okay, we have seen. What is this wall pressure? We have not seen. Wall pressure is a new terminology for us now. What is wall pressure? Associated with this. When the cell is turgid, when placed in an hypotonic solution, an opposite pressure is exerted by the cell wall on the cell. In response to the turgor pressure experienced by the plant cell, it will respond to consider this as a turgor pressure. Pressure experiences when the solvent outside the cell enters into the cell. In response to this pressure, plant cell will respond to it by another pressure called as wall pressure. Equal but opposite pressure. Pressure is inward in nature. This is called as wall pressure. Another terminology, what is plasmolysis? What is plasmolysis? When cell is placed in an hypertonic solution, we have seen that water, when cell is placed in an hypertonic solution, that is plant cell, okay, water moves out. The shell will shrink. This kind of condition is known as this shrinkage. This kind of condition is known as plasmolysis. Okay. This plasmolysis process is reversible. The cell can be reverted into normal condition when the same cell is placed in an hypotonic solution. The cell will become normal. In hypotonic solution, water from the environment will enter inside the cell when placed in a hypotonic solution. When a plasmolyzed cell is placed in a hypotonic solution, water from the environment will enter into the cell. Plasmolysis process can be reverted. Okay. Hope so far we have covered about basic things: turgor pressure, wall pressure, tonicity, plasmolysis and their characteristics. Tonicity, plasmolysis and their characteristics. Hope this end about the experiment with Osmosis. Hope this is very helpful for preparation of CSIR, NET, for GATE, for other competitive exams regarding to life sciences. Okay. Hope this is very helpful. In next series we will see about the components of water potential, gravitation, matrix potential, osmotic and pressure potential. Okay. Thank you.